We'll go ahead and get started. So we have a distinct pleasure. We have a guest faculty today. Ramana Sidhaye is a assistant professor in the Division of Pulmonary and Critical Care Medicine, as well as you're in physiology as well, right? No, just landed. So when we kind of try to think of evolutionarily, and this is good as you guys started scientific foundations, today, as we start to look at what the evolutionary advantages that allowed life to move kind of out from a single-celled organism to the complex organisms that you guys are going to take care of in 2 short years. One of the key things that has been part of that evolutionary step has been the development of epithelium, or the lining of kind of our vital tissues. And it was that interest that promoted the Pulmonary and Critical Care Fellow, Dr. Sidhaye, about 8 or 9 years ago, to began a career in investigation working with Dr. Peter Agre, who won a Nobel Prize for cloning originally identifying the Aquaporin water channel, and with Dr. Landon King, who's now actually the chair of the Division of Pulmonary. So Ramana chose to work with those 2 researchers to really look at epithelial biology. So when we say epithelial biology, this refers to, especially important in the lining of the lung and the sinuses, it refers to everything that determines the barrier that those lungs are. So it has relevance to problems where that barrier breaks down, such as acute lung injury, or when patients have go into the ICU and develop leakiness of their lungs. It has relevance to why our nose runs when it's cold outside, or when it's cold in the air conditioning inside. It's not cold outside here much. So Dr. Sidhaye began as an undergrad and medical student at Northwestern University. She stayed there for residency and then was chief resident there. And in 2002, her and her husband came to Hopkins, where she began her pulmonary and critical care fellowship, and we began to work together. She's a year ahead of me. She's gone on to become an assistant professor. She's funded by the National Institute of Health, by the Flight Attendants Medical Research Institute, and she's a great example of a true physician scientist, publishing basic science investigation into how epithelium, how those linings work, and caring for patients largely in the ICU, but also in pulmonary clinic as well. So she's going to talk to us a little bit today about an introduction to how epithelial biology is relevant to a couple diseases in the lung.
Thank you, Patrick. First of all, this is pretty informal. So as the people from Hopkins know, usually when I give a talk or anyone gives a talk, I'm interrupted before I get past slide 1. So feel free to interrupt with questions. If there's anything you don't understand, just raise your hand and we can go through. So the title of my talk is The Airway Epithelium and Environmental Exposures. If I can figure out how to use this. That's not it. And that's not it. Okay. Let me... Might be easier just to do this, the old-fashioned way. Oh, this is forward. Got it. Okay. So the airway epithelium, it's something that a lot of us don't think very much about. But the average person breathes about 8,000 liters of air a day. That's a lot of that the airway epithelium is exposed to. And it's the first line of defense against all the respirable environment. Despite this, despite the fact that it's sensing so much from the environment, there's really relatively few diseases that we associate with primary airway epithelial dysfunction. The most common one people think about when they think about airway epithelium and dysfunction is cystic fibrosis. This was a quote from the 1900s, actually late 1800s, where at that time mortality was extremely high in patients who, as they said, had a salty kiss from the brow. Right now in the U.S., 30,000 children and adults are affected with cystic fibrosis, 70,000 worldwide. And you talked a little bit about protein folding earlier today, but this is a defect in the CFTR gene resulting in problems in CFTR protein production, either in folding, in localization, or in function at the membrane, depending on the mutation. This defective gene and its protein product causes the body to produce very thick mucus. Now, CF is, as I said, what people usually think about when they think about airway epithelial diseases. However, there are some that are much more common. Actually, before I go into this, this is sort of what is wrong with the epithelium in CF. This is an electromicrogram. You see the cilia. It's surrounded by a periciliary layer of fluid. So this is a normal airway epithelium. It's actually not from human, but illustrates the point well, where you can see that the cilia are extended, allowing them to beat and clear effectively. Here, in a CF model, you see that that liquid layer is very much condensed, causing the cilia to be flattened. And this causes problems in clearance and other problems associated with cystic fibrosis. However, there are much more common diseases that actually might have epithelial origins. Asthma is an example of this. The WHO estimates that 300 million people suffer from asthma around the world. It causes problems in 250,000 annually in the US. It's the most common chronic disease amongst children in the world. It's actually found out recently. It's come as #1. And it's due to a combination of environmental and genetic factors. So what does the airway epithelium, what happens in asthma? We think of it as bronchoconstriction, constriction of the bronchus, which people associate with smooth muscle problems. But how is the airway epithelium actually altered? Well, it turns out that there are a lot of proteins that regulate the barrier function of the airway epithelium. So that means these proteins regulate how an airway epithelial cell interacts with its neighbor. Here's some proteins that are examples of, shown that are involved in this cell-cell interaction. They form in 3 basic junctions. They're called the tight junctions, which are closer to the apical or the top layer. So if you think of the airway here where the airstream is, this area is thought as apical, and this area is thought as basal or basolateral. Closest to the top, or apically, are tight junction proteins. These proteins, again, regulate solute and interactions between cell-cell contacts. The next, as you go further down, are adherence junctions. That didn't sound good. Adherence junction proteins, which also another layer to Is that better? Yeah. Here. Is this better? So adherence junction proteins are another example of proteins that interact with both the cytoskeleton skeleton of the epithelium. So these are the proteins that made up the backbone of the epithelium and extend out to cause changes in cell-cell contacts as well. And finally are the desmosomal proteins, which are towards the base, which also regulate epithelial interactions. Now, an investigator in Europe, at London, Holgate and his colleagues, did an interesting study where they looked at young children, ages 2 and younger, who presented to the ED with wheezing. They took, actually, biopsy, something which is very difficult to do in the US at least, of all these children, and then later on looked at retrospectively to see how many of these children went on to develop or be characterized as having asthma compared to others who just had some sort of viral infection and wheezing as a result. And what they found is that in these children who were 2, 3, something of that age, that patients who went on to having asthma had changes in these tight junction proteins that occurred so early on before they were having the multiple exacerbations associated with asthma. So it's actually now people are starting to recognize that maybe there's epithelial dysfunction that occurs in these patients that actually precedes the inflammatory changes that are associated with asthma. COPD, chronic obstructive lung disease. This is the 4th leading cause of death in the United States at the moment. More than 12 million people are currently diagnosed with COPD, and it's thought to be due to exposure to noxious particles or gas, most commonly worldwide from tobacco smoke, though other particles and gas, particulate matters, coal exposures, wood-burning stoves, other things are also associated with changes associated with COPD, and causes an inflammatory response in the lungs. So what happens to the epithelium with patients with COPD? Here's an H&E stain of normal epithelium. This is something you guys will be seeing more in time to come. But you can see there's a very regular pattern in the epithelium and the changes in how the epithelial cells interact with its neighbors. This, just to introduce the concept to you, is called a pseudo-stratified epithelium, where all the cells interact with the basement membrane. In a COPD bronchus, you can see that there's changes, dramatic changes in the epithelium, and this is an example of goblet cell hyperplasia, increasing in expression of cells that express mucus. And this can change to a point where it causes a stratified epithelium, where all these cells don't necessarily touch that basement membrane, and it changes a lot in terms of how it interacts with its neighbors and epithelial function. Ugh. Okay, so I said we're exposed to 8,000 liters of air a day. So what we breathe in and what our epithelium senses is very much dependent on what's around us. So the very act of breathing in creates shear stresses, stresses that are sensed by airflow over the epithelium. In addition, if you breathe environment here with particularly polluted areas, or if you're sitting next to someone who happens to be sneezing and releasing out various infectious particles, all these particles are then result in inflammatory responses. So how does the epithelium respond to these luminal stimuli? You have a luminal stimulus, whether it's just the act of airflow or some particle in the air. And the epithelium can do several things. It can secrete proteins. Again, this is the lumen, or where the air is. So it can secrete things apically to the areas of the lumen. And these are defensins, things to combat infection, to combat inflammation, cytokines and chemokines to actually recruit cells to allow for inflammation, and mucus and mucins. It can send off signals to subepithelial tissues. These are tissues below the basement membrane, such as the smooth muscle cells or to the vasculature. It can give stimuli to its neighboring cells or to itself in an autocrine or paracrine fashion, and result in changes in signaling proteins, in ligands, and other cytokines. And finally, it can alter the way it binds or interacts with its neighboring cell. And this interaction or binding between cells is where the focus of my research has been. So if an epithelial cell interacts with its neighboring cell more tightly, we call it barrier enhancement. So the barrier is stronger, and that's associated, or that causes decreases in paracellular permeability. Paracellular permeability is very specifically the permeability between cells. So there's ways to think, what's the use of paracellular permeability? Very crude thought, or basic thought, is that if you have a tighter barrier where there's less paracellular permeability and you have exposure on the apical side, much less gets through to the basolateral side. So less gets exposed to basolateral ligands or receptors or basolateral tissues as a whole. However, if you have a leakier epithelium or increases in paracellular permeability, those things in the lumen can get through to the basolateral side. So why do we have this ability to adjust the permeability? One possibility is that we could have just cement on our airway epithelium that would segregate everything in the lumen away from everything inside of us, and we should be protected, right? Well, it turns out that we actually have a very fine-tuned system. In times when you have a very tight epithelium, decreases in paracellular permeability, sometimes you have ligands that are sent out to the luminal side. So these are proteins that are looking for their receptor to bind to. And our body has developed systems such that occasionally the receptor is located on the basolateral side. So if you have a very tight epithelium, that ligand can never come in contact with its receptor. However, if you have increases in permeability, you can have ligand-receptor interaction, where that protein comes in contact with its receptor, resulting in various epithelial stimuli. So what my lab found is basically under several conditions you have this separation of ligands and receptors, and changing epithelial permeability results in altered epithelial function because you regulate how well the ligand comes into contact with its receptor. For example, in response to stimuli such as osmotic stress, cyclic AMP, you see releases of the EGF receptor ligand, neuregulin. It's actually tethered to the apical side, and in response to these stimulus, it's cleaved. And if you have adequate paracellular permeability, you can have receptor-ligand interaction. Similarly, interferon gamma, which sends off a bunch of inflammatory stimuli, is located on the apical side. And in response to certain stimuli, in a condition where you have adequate paracellular permeability, that receptor and ligand are allowed to interact. Turns out it's not unidirectional, but in fact that permeability can go both ways. So substance P is a substance that's secreted from the NANK system, which is the vasomotor system that's located in the basolateral space. And substance T's receptor is located on the apical surface. So only under conditions of increased permeability do that receptor and ligand come into interaction. So the epithelium can modulate the space or the interaction between neighboring cells and therefore dictate whether a receptor can come in contact with its ligand. And that can lead to a bunch of different stimuli. Now, one thing that my lab is now exploring further is it turns out that via some of these signaling pathways we just talked about, and by other ones that I have not mentioned, that in addition to just dictating receptor-ligand interaction, these changes in permeability can also affect the way the epithelium works as an innate immune organ. So the epithelium has the ability to present several different receptors, ICAM1 being an example, that allows white cells, neutrophils, to migrate through the epithelium and to go to sites of infection. But based on how tightly the epithelium interacts with its neighboring epithelial cell, it can dictate how its response to some particles such as viruses. So for example, you come in contact with certain viruses, and in a very tight, impermeable barrier, you have a certain amount of expression of epithelial ICAM as well as chemokines. Chemokines are proteins that recruit other inflammatory cells. So when you have a very tight epithelium, it turns out that it seems like we have a very dampened signal to recruit other cells to that milieu. However, that's just the recruitment of the cells. Under conditions where you have same exposure, increased epithelial permeability, it turns out you have increases in signals to try to get those neutrophils both to the epithelium and across the epithelial barrier in order to potentially combat infection. So the way that the epithelial cell interacts with its neighbors also seems to dictate the way that cell interacts with other inflammatory cells. And this is sort of an area where my lab is now looking at in terms of research, trying to determine molecular mechanisms that dictate these changes in interactions. So a summary up to this point is that the airway epithelium dynamically regulates paracellular permeability in response to both physiologic and pathologic conditions. We've looked at a bunch of different things, from just things like cyclic AMP, changes in osmotic stress, and changes in shear stress in terms of the physiologic conditions to see that there's regulation of the epithelial barrier function and changes in cell-cell interaction. While we were looking at molecular determinants that determine this evidence of how cells interact with each other, we came across two relatively novel proteins that seem to dictate how the epithelium relates to its neighbors, proteins that weren't really considered in this pool. Aquaporin 5 is one of them. Aquaporin 5 is a water channel, so it's a tetramer. We talked a little bit about earlier today about proteins interacting with other proteins and forming monomers and multimers. So it's a tetramer, so 4 aquaporin proteins come together. Each of the proteins have its own central hole, so it doesn't form a central hole between the 4 proteins, but each of the 4 proteins allow for the transport of water through it. In response to physiologic changes in shear stress, you see an internalization of aquaporin 5, leading to aquaporin 5 degradation and changes in paracellular permeability. Try to sort of simplify how we look at things in terms of how do we measure paracellular permeability in our experimental protocol. So we grow airway epithelial cells on inserts. So that allows us to separate out what we call the apical layer and the basolateral layer. So the apical layer is exposed to air, just as it is in our airways, and the basolateral layer is exposed to media, similar to the bloodstream and all the other nutrients that it gets in the airways. This allows the cells to differentiate, form cilia that beat, produce mucus, and act very much like epithelial cells do in our airways. And what we do then is stimulate them in response to whatever we're looking at, and then we measure what the barrier function is. As I mentioned, barrier function is what we're referring to is specifically how molecules move between cells. So we look at a large cell-impermeable protein, dextran, which doesn't go through the cell but only goes between cells, and then we label it to something that fluoresces so we can just detect it. And we add it to the apical side, we measure it from the basolateral side, and if we see more fluorescence, that means more went through, and if we see less fluorescence, that means less went through. So FITC dextran is what we look at to see what the permeability is. And when you look at just airway epithelial cells by themselves, you see some level of permeability, a baseline level of permeability. Some FITC dextran goes through. If you expose them to, you add GFP, cause it to induce GFP expression, which is just a control, doesn't really do much in this state, you don't see much change in permeability. When you overexpress aquaporin 5, so you're adding aquaporin 5 to this, you start seeing a significant increase in paracellular permeability. And in response to shear stress, you see a decrease both in aquaporin 5 protein, which I don't show here, but also a decrease in permeability. So our data suggests basically that the ability to regulate aquaporin 5 allows the epithelial cells to regulate paracellular permeability, which was surprising since it was primarily thought to do with transcellular water transport and not paracellular permeability. So this is aquaporin 5 protein. We kind of talked about protein structures. Here's the C-terminus of aquaporin 5, which is intracellular, is the intracellular portion. It's a pretty small protein. Right here in this area, there's a beta-pleated sheet, something you guys talked about briefly this morning, with various sites that can be phosphorylated or other post-translational modifications. And what we did was we purified the protein, allowing it to fold the way it would conformationally, and just mixed it with purified microtubules. We were trying to see how a protein on the apical surface would alter something that occurs between cells on the lateral surface. So what happens that changes something that's sitting on top of the cell to cause a cell to interact laterally that's different with its neighbor with and without this protein. And after looking at many things, we started looking at the cytoskeletal proteins and came across microtubules. And what we found is that if you add purified aquaporin 5, which a bunch of microtubulin, so the monomers that form microtubule chains, and you just add it together with some juice to make them happy, and look to see how much microtubule formation you get. With purified aquaporin 5, you see increases in microtubule formation. And that's in the red. So this is with a control that we know, a drug that we know causes microtubule formation. Down here is with a drug that we know inhibits microtubule formation. Here's just controlled tubulin by itself, and if you add aquaporin 5 to the mix, then you see increases in microtubule formation. If we add aquaporin 1, a related protein, but not exactly the same, and the C-terminus is a little different between the two, we don't see that. So it's very specific to aquaporin 5 that we see changes in microtubule formation. And then what we did was we cloned just the C-terminus. So all the part that I showed you that was within the cell, we cloned that, purified that, and added it. And it turns out that's all you need to allow for microtubule formation, just that C-terminus, which actually has nothing to do with the part of the protein that allows for water transport. So, so somehow these changes, this Aquaporin 5 results in changes in microtubule polymerization directly, because this is in a cell-free system with nothing else except for the purified protein and purified tubulin. Another protein that we came across that seemed to alter barrier function was septin-2. Septin-2 was identified in yeast, and it's known to separate the cytoplasmic contents between the mother and daughter cell. And what was known about it is that it was involved in apical sorting. However, really what its role was in airway epithelium, which are non-dividing in human cells, was completely unknown. So we started looking at it, and what we found that is in response to shear stress, where I described that there was enhancement in barrier, we saw that it moved out to the membrane compared to intracellularly, where it is under static conditions. And in addition, and I won't go into all of this, but this is basically actin. It's part of the actin cytoskeleton. We found that if you altered septin-2 expression, you alter actin dynamics. So right now we have a working model where in response to shear stress, we get a decrease in Aquaporin 5, results in microtubule depolymerization. So more Aquaporin 5 causes more microtubule polymerization, less causes microtubule depolymerization. That same stimulus causes septin-2 to go to the membrane and causes increases in cortical actin, and somehow that seems to translate to changes in cell-cell interactions. So that's where our lab is at this time, where we're trying to figure out the molecular mechanisms that tie the microtubule polymerization to the actin cytoskeleton and how that translates to changes in cell-cell interaction, which we think occurs by a specific protein called E-cadherin. So I talked about some physiologic stresses, shear stress, and mentioned cyclic AMP and osmotic stress earlier. But earlier on, I started the lecture saying that we have a lot of exposures. We're exposed to particulate matter, we're exposed to other things in the environment. So how do these things affect the way our epithelial cells respond to what we breathe in? So I said, we are exposed to particulate matter. Nowadays, we're exposed to environmental nanoparticles. These are smaller sized particulate matter, which are increasing in the environment because of the industry use, and as well as other infectious agents, which I won't talk about as much in this lecture. So airborne particulate matter, dust, for lack of a better word. There's many sources, both natural and man-made, but it's basically all the stuff that we consider pollution in the environment. It comes in a variety of sizes. People have looked at it very specifically and found that these micron-sized fractions, 2.5 micron and the 10 to 2.5, both that form in the coarse and fine fractions of particulate matter, seem to cause changes in human health. So increases in particulate matter in the urban areas are associated with increased mortality, increased ER visits, increased hospital admissions for cardiovascular and respiratory disease, and can occur as quickly as 0 to 3 days after exposure. So these are things that we're dealing with routinely. This just shows that based on the size, there have been associations with different types of symptoms, from wheeze, nocturnal symptoms, limited speech, need for rescue medication. So how did I incorporate this to my cell culture system? Basically, we have a collaborator who collects the environmental particulate matter using concentrator and can look at the specific size fractions. And so we look at particulate matter 2.5 micron size. And we expose the apical surface of the airway epithelial cells to this. I was describing how we grew the cells on inserts, and here's a picture of the inserts. These are the cells that are located here. The apical surface is exposed to air. The basolateral has media, as I described before. And we just basically throw on a bunch of dust on top of these cells and see what happens. Turns out, something that I was really surprised by, when you throw on dust and you look at the barrier function, dirt enhances the epithelial barrier. So when we looked at the fit-C-dextrin, the permeability between cells, after the exposure to particulate matter, there was an enhancement in barriers. So the cells were interacting with this neighbor even tighter than they did before. And it's only after an exposure to a second dose did you see increases in paracellular permeability. I don't think this projects well, but basically what we noticed is that in response to one dose of particulate matter, you see an increase in membrane expression of septin-2, I mentioned this protein before, and changes in cortical actin suggesting mechanisms why there would be an enhancement of barrier, And after a second exposure, you don't really see these changes, suggesting molecular mechanisms why there might be changes causing an increase in permeability. In addition, Aquaporin 5 gets internalized after a first dose of particulate matter, but then starts going back to the membrane after a second dose. So I talked about these micron-sized particles. Now, is the size matter? I specifically started talking about micron size, but does changes in nanoparticle exposure or smaller exposure cause different effects on epithelium? So when we talk about nanoparticles, we're typically referring to particles less than 100 nanometers in diameter. That's what's typically used in the industry. And there's been, as I mentioned, an increase in industry uses resulting in much more airborne exposure. Aside from that, there's been a lot of interest in using these nanoparticles for medical therapeutics and theragnostics. Studies suggest that somehow the tissues seem to react differently to these particles than they do to larger micron-sized particles, and there might be even more inflammation and potentially more susceptibility to forming cancer or tumors. And what people don't know, and what remains to be discerned a little bit out there in the industry, is what the effects of the physicochemical properties of the nanoparticles are on human responses. So does it matter whether we use positively charged molecules or negatively charged molecules, or does it matter what the specifics of the size or shape are? Do we respond differently? And these are all important because without any of this knowledge, this use has increased tremendously in the industry, but we don't really know how we respond to them. So our first question was, do these nanoparticles even cross the barrier? So there's some thoughts that because of the way we breathe, that the epithelial cells, that they will be trapped within the mucus area, and they never actually cross the barrier. So we have a cell system that actually produces mucus and has cilia that beat, so it's an ideal system to look to see, at least in our cell culture model, whether it crosses the epithelial barrier. And it turns out they do. But based on the charge, they cross at different rates. So here's, we used these nanoparticles, which are called quantum dots. They're often used in both medical and as well as industry uses, but their properties that they autofluoresce. They're easily trackable. So we exposed the apical surface of our cells to these quantum dots and measured it from the basolateral surface to see if it went through or not. And both negatively charged and positively charged particles cross that epithelial barrier, and the positively charged particles seem to cross faster than the negatively charged particles. So then we wanted to know whether there was anything about the way they crossed. So they both seem to cross, but is there any difference? And is there any way we could track that whether they cross the same way or not? So we're just starting these studies. But I don't know how well you can see from out there. The green is a membrane marker, so you see the outlines of the cells. And here in the red are the particles, and you can see large conglomerates, and they're actually, when you see the co-label, they're within the cell. However, it's hard to project here, but basically the negatively charged particles seem to go between cells. So the charge of the particles dictate the route which the nanoparticles are crossing the epithelium. The next question was then, does it affect the barrier? Well, do they just passively cross, and do they do anything to the epithelium, or do they cause it to alter barrier function? And in fact, we found that that we do see changes in resistance. So I described permeability as a way to look at large particles moving through the epithelium. Another way people look at permeability is by measuring resistance of the monolayer. So if you have very tight interactions between the cells, you think you might have if you have a very tight interaction, you might have an increase in resistance and a low permeability, and vice versa. However, what we found was that you see a drop in resistance in this epithelial monolayer, despite whether we use negatively or positively charged particles. Now, resistance, while it's commonly reported in the literature as a marker of permeability, is a little tricky because the passage of a positively or a negatively charged particle itself might alter resistance, and it has nothing to do with barrier function of the epithelium, but more with a positive particle that's allowing for conductance of electricity within there. So we looked at also phytidextran, and we found that these positively charged particles caused an increase in barrier function, or rather a decrease in paracellular permeability compared to negatively charged particles. So not only do the particles move differently, they also cause differential effects on the airway epithelium. Now we're trying to see how these particles then affect these proteins that we were talking about. And studies are preliminary, but we see already that the negatively charged particles cause changes in proteins more dramatically than we see in positively charged particles. We're further working out these studies right now to determine what the effects of particles are on both barrier function as well as cell-cell contact and proteins that regulate it. Finally, this is just a slide to show that I talked briefly about how the direction of our lab was now that we're looking at if a cell, an epithelial cell, interacts with its neighboring epithelial cell differently, does that mean that it's going to interact with an inflammatory cell or immune cell differently as well? One of the ligands that the epithelium presents is ICAM1, and that recruits neutrophils to the epithelium and allows it to interact. And what we see is if you alter the epithelium by knocking down Aquaporin 5, you see a difference in ICAM1 expression. Now, Aquaporin 5 can do a lot of things. One of the things we think it does is alter microtubule dynamics. And if you treat the cells with another drug that basically causes microtubule depolymerization, you see similar differences in ICAM1, suggesting it could be that Aquaporin 5 is doing this by cytoskeletal manipulation. So just to summarize, I talked about a lot of things, but airway epithelial dysfunction is likely contributory to some significant diseases worldwide, including asthma and COPD. Dynamic changes in paracellular permeability can affect epithelial signaling. Both physiologic and pathologic signals alter paracellular permeability, and inhaled micron and nano-sized particles can contribute to these changes. So there's still a lot to be worked out. We're still trying to determine how the epithelium responds to other sized particles and exactly what happens with the cell-cell contacts. And that's in non-infectious particles. But there's another field that we're emerging looking at in terms of bacterial responses and viral responses. And frankly, all of this remains an open question. I want to thank the members of my lab, Eric Chow is my technician who's done most of this work, and my postdoc, Srinivas, Landon King, my friend and mentor, and a lot of my collaborators, Patrick Bricey, who provides us with particulate matter, Peter Searson and Denise Wirtz, who are nanoparticle experts, and I've been collaborating with them with the nanoparticle studies, Larissa Shimoda and Mike Katerina, who are calcium signaling and TRPV4 experts, which I didn't talk about, but that we look at in terms of pathways that regulate Aquaporin 5. Thank you.